We'll also talk about the inflammatory process, which should be reviewed, but we'll go into a little bit more detail. And we'll identify several inflammatory chemicals and indicate their specific roles. We'll name the body's antimicrobial substances and describe their functions as well. All right, so the function of our immune system is defense against bacteria, which is the first picture, viruses, which is the second picture, fungi, also defense from grafts and transplants. Even though we don't want transplants or grafts to be rejected, it's the immune system that protects us from it because they think this is something foreign, it's an invader, I need to attack it. So your immune system would be responsible for rejecting grafts or transplants. And then also from cancer. What kind of cancer is that on that picture? Melanoma, which you should have learned in Bio 32. All right. So how our immune system is divided, we have what are called innate defenses and adaptive defenses. And there are some description of these in your lecture notes. The innate defenses, they are called innate because they're present from birth mostly. Some of these develop like in the first few months of your life, but most of what's in this category is present from birth. It's very non-specific. And so it's kind of like, this is going to be your first and second line of defense. You won't get any memory or specificity until you get to the next category. So there's no memory cells associated with your innate defenses and no antibodies that are produced by these innate defenses. There's very non-specific, and they are there as your first and second line of defense. So your first line of defense is going to be your surface barriers. So this is going to be your skin and your mucous membrane. So the things that are on the immediate outside of your body. So your skin and your mucous membranes line any entryway into the body. So your digestive tract, your respiratory tract, your reproductive tract are all lined with mucous membranes to be able to trap any bacteria, viruses, or any foreign invaders that may come into those entryways. So if those fail to protect you from the foreign invader that's trying to come in, then your second line of defense are the internal defenses. So these guys are external. And then your second line of defense is internal. This includes phagocytes. You don't have to write all these down. We're going to go over each one of these individually. But we have phagocytes that are going to do phagocytosis and engulf things that don't belong. We have natural killer cells, which you haven't heard about before in this class, so we'll go over those. Inflammation, which should be a review for you all, some different antimicrobial proteins, and then also fever. Okay, so those are all part of your internal defenses that are part of the innate defenses, the things that you are born with. And again, we'll get into all of these individually as we go on the lecture. Your third line of defense is going to be your adaptive defenses. These are very specific defenses, so they are activated on a very specific strain of a virus or a very specific species of bacteria. So they are targeting things that are very specific, and they are acquired over your lifetime. So you're not born with these. You develop them as you get exposed to different things over your lifetime. So this includes your humoral immunity, which is going to be mostly antibodies. We'll talk about that a little bit later. That's going to be carried out by your B cells. And then cellular immunity, where cells attack those things that are foreign, and that's going to be your T cells. Specifically your cytotoxic T cells, but helper T cells will help get them activated. And these are also going to be internal. So it's just a quick overview of your innate versus your adaptive defenses, but we'll go over each one of these individually. Questions about these, though? Yeah, Susie.
The first one is non-specific, and the second and third are specific?
Yes, they are specific. So, well, no. Your first and second are non-specific. So everything in this category is non-specific, and you're born with it. The only ones that are specific and will have memory are going to be your adaptive defenses.
Okay.
So your first line of defense are going to be your surface barriers, like your skin and your mucous membranes. We call these constitutive, which means that they're always on. So this defense is always on. It never takes a break. It's always protecting you. One thing about your skin and mucous membranes, they're only going to be able to protect you if they're intact and not damaged. Okay, so make sure that when you describe your skin and mucous membranes as a first line of defense, they have to be intact. Okay, because as soon as you have an injury to your skin, now you're vulnerable to something entering the body. And so it's no longer going to be a good defense if there's damage to it. So it has to be intact skin and mucous membranes. So we have actually 3 different categories of our first line of defense, these barriers. So the ones that were listed on the diagram that we saw before, and I'm going to write a list out for you all. And there's some blank spaces in your lecture notes to write these out. But the one that was on your list are your mechanical barriers, which are your intact skin and mucous membranes. But then we have other chemical and microbial barriers too that are going to help us with this first line of defense. So chemical barriers include acids. So you have something, and you don't have to write this down, I just want to give you examples. But you have something called an acid mantle that is on your skin. So there's a very, very thin microscopic layer of acid on your skin that inhibits or slows down the growth of bacteria. So it makes it harder for bacteria to grow because your skin is slightly acidic from that acid mantle. Same thing for females. The female vagina, that birth canal, it also has an acid mantle to discourage bacterial growth, so it protects you there a little bit. Your gastric juice in your stomach is very, very acidic and protects you from bacteria in your gut. And then your urine as well is slightly acidic, which helps keep bacteria away from your urinary tract. Obviously, it doesn't always work. Sometimes you'll get a urinary tract infection, which we'll talk about after exam 2. But it is slightly acidic to try to discourage that bacterial growth. Enzymes as well act as a chemical barrier for us. We have what are called lysozymes, which you guys remember lysosomes from B32. Well, they're an enzyme that's within it called lysozymes that you find in your tears and in your saliva that help to break down bacteria if you come into contact with them. Pepsin also is an enzyme that can help protect you from bacteria as well that lives in your stomach. Another chemical barrier is mucin. What are the glands that are responsible for making mucin? They are a unicellular exocrine gland. So a single cell that makes mucin that's used to make mucus that you learned in Bio 32. Goblet cells. So goblet cells produce mucin, which is a protein that helps make mucus. Mucin plus water makes mucus. And that mucus we find in our digestive tract, we find it in our respiratory tract, it's within our saliva, and that's going to help trap any bacteria, viruses, debris, anything that doesn't belong gets trapped in your mucus, and then hopefully you spit it out. Okay. Also, what is the oil produced by your sebum? That also provides some protection as well. You also have a antibiotic that your most prominent sweat gland produces called dermacidin. What is your most prominent sweat gland out of the 2 that you learned in Bio 32? The eccrine sweat glands. Those are the ones that turn on by changes in temperature. What was the other kind of sweat glands you learned about? Apocrine. And those get activated by stress and embarrassment, things like that. And I think that's it. There are more, but these are just some good examples, especially things that you've learned before that we can reintroduce now. And then the last barrier is going to be also your microbial barrier. So we all have good bacteria on our skin and in our gut that helps to fight off bad bacteria. We have what's called a symbiosis with them. They feed off of the chemicals on our skin or the chemicals within our digestive tract, and in return, they protect us from bad bacteria. So those are all part of our first line of defense, all the barriers that foreign invaders have to get past in order to try to infect us. Okay. Any questions about these barriers? Who's still writing this stuff down? Okay. The good bacteria keep the invaders or the bad bacteria out. We do have some bad bacteria that live within our gut. Everybody's makeup of their bacterial flora is different. But we do have some bad bacteria that live within us, and the good bacteria keep the bad bacteria at small numbers, so it doesn't make us sick.
Professor, you said dermacidin was what?
From ecrin sweat. Yeah. So it's an antibiotic that we naturally produce. All right. So again, this is going to be our mechanical barrier, the skin. This is part of our chemical barrier. That's the mucous membrane of the respiratory and digestive tract. And then here is our microbial barrier. You can see the probiotics are the good bacteria. They're the ones eating all the food and leaving nothing for the bad bacteria so they can't grow. All right, so those were our surface barriers. We're going to talk about internal defenses next. So this is still the innate defenses. These are still the ones that are non-specific, still the ones that we are born with that do not have any kind of memory. They're just there to fight off anything that does not belong. So this includes our phagocytic cells and our natural killer cells. So our phagocytic cells are like Pac-Man, they just go around and eat everything. That includes like our neutrophils and our monocytes and macrophages. Remember, those are the same thing, monocytes in the blood, macrophages in the tissue, but those are our phagocytic cells. Then we also have natural killer cells as well. So we'll talk about those first. But then we also have inflammation, fever, and antimicrobial proteins, and we'll get to all those things. But we're going to talk about the cells first. So our neutrophil, which is this blood cell here, and then our monocyte or macrophage, which is that cell there. Don't forget to review the blood cells because those will be on the next exam as well for the immune system. So if you're good on those, then you'll have another chance to get 3 questions right. So phagocytes. So phagocytes include our neutrophils and macrophages. Or monocytes. It all depends on what their location is, what we call them. But what do neutrophils fight off? Bacteria specifically. These are the only ones that are part of our innate defenses that are kind of specific. They can fight off other things as well, but their main target is bacteria. Monocytes and macrophages. You should know these are more aggressive because they really will just eat anything that doesn't belong. So they're going to be much more aggressive in their phagocytosis than neutrophils are. So you should have already learned a little bit about how phagocytosis works. Phagocytes are going to adhere to whatever the foreign invader is. This may be a bacterium. And then they form these little vesicles They're first called a pseudopod, but you don't have to know that. But they're eventually going to form a vesicle. And that vesicle is going to, which is called a phagosome, it's going to merge with a lysosome, which you should already know from Bio 32 has digestive enzymes. And so this phagosome, which is a phagocytic vesicle, and a lysosome merge together to release those enzymes onto the foreign invader. When they do merge, they merge into something called a phagolysosome, which is just the pushing together of a phagosome and a lysosome. And so the enzymes are released on that foreign invader, whether it's a bacteria, virus, or what have you, and the enzymes will break that down. And then once we break that down, any parts that can be recycled and used by the cell, it will, and then anything else will get delivered out by exocytosis. Okay, but you all should be familiar with that process. Okay. Any questions about phagocytosis? Okay, and you can see here's a macrophage here. It can reach out its cytoplasmic extensions and grab things in its environment that doesn't belong, that it sees as an invader or something harmful. Okay, natural killer cells. These are found in both the blood and lymph, and this is all, I think, or wait, is it your notes already? Okay, so write down these notes here. They are found in the blood and lymph. They are an aggressive lymphocyte, so they're a specialized lymphocyte. They target things that are nonspecific. So essentially they are targeting cells that are not acting normal, so our own cells that are not behaving properly. And so this includes cancer cells, which are growing at an abnormal rate, and then virus-infected cells. So any cell that's not acting like it should, a natural killer cell may fight against it. They are non-phagocytic. How they work is they induce apoptosis. So they attach to their target and they release chemicals that cause the cell to implode. Now, one of the things that unfortunately a natural killer cell can kill or harm is an embryo. So you can see here's a natural killer cell attacking an embryo. An embryo would be a self cell that is not acting normally because it's, when somebody gets pregnant, this is a new event. So natural killer cells are just not familiar with what's happening. They see it as a foreign invader and they start to attack it. Okay, this can happen and this would cause a miscarriage. Oh, I don't like that color. We'll do that.
Is that common?
I mean, miscarriages are common. To know that it's caused specifically by natural killer cells, they would have to do more testing to figure it out. So I think that number is maybe higher than what we think it is. But it can happen if we have too many natural killer cells. But what's really interesting is that research shows that natural killer cells are needed for a successful pregnancy. So we need natural killer cells for the first part of pregnancy and development because they release some certain chemicals that help with the process. But if we have too many of them, then they can be overreactive and attack the embryo itself. So there has to be a balance in that. Okay, any questions on natural killer cells? Okay. Anybody still writing these notes down? Okay, I'll wait. All right, so another part of our second line of defense is going to be the inflammatory response. This is going to stop the spread of agents or foreign invaders to nearby tissue. It cleans up any cell debris and pathogens, and then helps with tissue repair whenever we have an injury. So you should know about this process. You learned about it in B32. We talked about it with blood and blood vessels as well. But first you have leukocytosis. Neutrophils enter the blood from the bone marrow. Then you have margination, which we didn't give it a name before, but this is where we have those molecules that slow down the white blood cells so they know where to get off when there's been an injury. Do you guys remember what we called those molecules? Cell adhesion molecules. So margination is the neutrophils or leukocytes getting stuck to those cell adhesion molecules and knowing where to stop and knowing where the site of injury is. So that process of them sticking to the cell adhesion molecules is called margination. And then you know about diapedesis. That's how they squeeze out of the capillary to get to that site of injury. And then chemotaxis, where when you have an injury to the skin, there are chemicals, inflammatory chemicals that are sent out, and that attracts neutrophils to that area as well. So the margination slows them down, but ultimately it's going to be the inflammatory chemicals that attract them exactly to where the injury is. Okay. Any questions about the inflammatory response? So we'll go into a little bit more detail about exactly what is happening. So we'll go through each line here. This is just a summary of what's going on. Now, what's in your notes is this here. This says the same exact thing. Okay, same events. It's just a little bit summarized into these little boxes. But what's happening is here you have tissue injury is what happened first. Okay, whether it's a nail or a piece of glass or whatever it is that penetrates your skin, that tissue injury is going to cause the release of inflammatory chemicals like histamine and other things that we're not going to get into, histamine being the one that causes permeability and causes the release of fluid. But these inflammatory chemicals, one, are going to cause arterioles to dilate. Okay, so they're going to get more permeable and more leaky. And because they dilate, that's going to allow them to hold more blood. So you're going to have what's called local hyperemia. We're just going to get more blood flow to that area because the vessels are dilated. So increased blood flow to the area, which is going to cause both redness and heat because of more blood localized to that area wherever the injury is. And then the heat, then increase in temperature, causes cells to work faster, increases metabolism, which leads to the healing of the tissue. The faster cells can work because of the higher temperature, the faster they can heal the tissue. Also, what's going to happen, these inflammatory chemicals can directly cause pain, which is going to cause possible temporary impairment of function. So you may not be able to move that particular site, depending on how big the injury is. So you may have some immobilization, which, I mean, not moving something that's injured is going to help with the healing processes also. This is going to cause the inflammatory chemicals also cause increased capillary permeability. So again, the vessels become leaky and fluid from inside the vessels, particularly plasma, is going to be excreted out, is going to escape out. Now, it is pretty much plasma, but when it's like this during the inflammatory process, the fluid that gets leaked out, we call it exudate. And so this is going to be leaked protein-rich fluid, this exudate here. That's going to cause swelling. The swelling could also cause immobility. The more that a part of the body is swollen, the less you're able to move it around freely. But again, that immobility is going to help lead to healing. Part of what's going to be released with that exudate is going to be clotting proteins, which is going to help with creating a clot and a scab in order to heal that tissue. Okay, so one of the big things to know is that all these things that are happening are all very, very necessary for the tissue to eventually heal from that injury. And also know when you have that swelling from the exudate coming to that area, swelling is then going to push on pain receptors. Do you guys remember what pain receptors are called from B32? Nociceptors. So the swelling is going to push on nociceptors, which is going to cause some pain. And again, that can lead to immobility. These 4 signs here, these are 4 signs of inflammation. So heat, redness, pain, and swelling are all signs of inflammation, and they're all necessary for the tissue to eventually heal. So some of the other chemicals that get released during a tissue injury is leukocytosis inducing factor. So basically it's going to attract white blood cells to the area. So you have leukocytosis, increased number of white blood cells in the bloodstream. Leukocytes migrate to that area. They marginate or get stuck to the walls at the site of the injury. They squeeze out of the capillaries at the site of injury. Then once they get into the tissue, they can then do phagocytosis on any pathogens or things that don't belong. Also dead cell debris. You just created an injury or you were just injured. So there's going to be some cells that are dead, some cell debris from those dead cells. And phagocytes are going to clean that up. And then area is now cleared of debris. Pus may form depending on the level of the injury. But again, all of that is going to be necessary for healing. So any questions? I know it's a lot of information, but as you study it line by line. This is what's happening. This is the effects. This is the sign of inflammation. And again, make sure you know it's all necessary for the tissue to eventually heal. So any questions on that process? Okay. And again, this is just a summary of everything where you have, and it just summarizes everything here. Arterial dilation is going to cause, because you have more blood to the area, is going to cause heat and redness. You're also going to have extra nutrients brought to the area, and that's going to be helpful for repairing and mitosis. This localized chemical release is also going to help with capillary permeability. So you get clotting factors within this exudate that gets released. The clotting here traps pathogens, and we're going to get pain and swelling, which may cause immobility. But again, this is necessary for healing. And then you'll also get chemotaxis, where you get the white blood cells to the area of injury to get rid of pathogens, get rid of any cell debris. And there will be inflammation that comes with that and swelling. But again, this is all necessary for healing. All right. So another part, this is still your innate defenses. They're still the ones that are nonspecific and what you're born with. But this is going to be an antimicrobial protein called an interferon. We have different types of interferon, but you can just call them interferon as a group. They are produced by viral infected cells to signal neighboring healthy cells that there is a virus around. And what it does, it slows down the viral spread in tissue so that white blood cells and antibodies can catch up to kill the virus. So how it works, what you're seeing here is here's a virus. Okay, it enters the cell and starts making copies, which can then possibly go and infect a neighboring cell. Okay, but what ends up happening when the virus enters the cell, it causes a gene to be activated within the cell, which causes the cell to then do transcription and translation to produce something called an interferon. That interferon then attaches to a neighboring cell, and now that neighboring cell, because of this interferon attaching to the cell membrane of this neighboring cell, causes the neighboring cell to produce an antiviral mRNA, which then blocks the entry of the virus. Okay. So basically what's happening is that this cell is going to eventually die and undergo apoptosis because it's infected by a virus. But what it does before it dies is that it just alerts all the cells around it. Like, hey, there's a virus coming around, protect yourself, produce these antivirals so that you don't get infected as well. Any questions about that? Isn't that cool?
Yeah.
They're like, it's too late for me, but save yourselves.
Right. Very polite.
Yeah. There's actually some treatments where they're pre-made interferons are given to hepatitis patients to help them produce antivirals to help protect them against the hepatitis virus. All right, the next antimicrobial protein are what are called complement proteins. This is a group of 20 proteins that work together. The complement proteins, again, group of 20 proteins. And they have many different functions in the immune system. But one of their functions is to produce something called a MAC. MAC stands for membrane attack complex. So how this works is that these group of complement proteins actually come together and create a pore in their target cell. So you can see that here. This is usually bacteria that they do this with. But again, they have many other different functions and can help assist the immune system in many different ways. But one way that they attack bacteria is that they create these pores. And what ends up happening is that water then enters those cells. Their cell contents leave the cell, and the cell basically implodes, okay, from the water rushing in. So it creates these pores all over the cell, water rushes in, and the cell implodes, or the bacterium implodes. So any questions about a membrane attack complex? So just, all you have to call them are just complement proteins. You don't have to know the specific names of any of them. I know they're numbered here. You would learn that in like an immunology class if you were to take an immunology class later. But a group of 20 proteins, they come together to create a pore in the membrane of bacteria. Once they create that pore or many pores, it causes water to rush into the cell, and then the cell will implode. And then the last thing that's part of our internal innate defenses, again, non-specific, no memory, is fever. So when white blood cells detect foreign invaders, they release a chemical called a pyrogen. Now, one of the functions of your hypothalamus, besides releasing hormones that you learned about, but you learned in Bio 32 that they also set our core temperature. And so when white blood cells sense a foreign invader and release these pyrogens, those pyrogens eventually make their way to the hypothalamus, and then the hypothalamus increases our core temperature. It sets our thermostat a little bit higher. Okay? And what this is going to do, a higher core temperature when you're sick, inhibits microbial growth. Okay, so it can inhibit the growth of bacteria and certain viruses, which is why you get a fever when you get infected. So the white blood cells tell the hypothalamus to increase the temperature because lots of different bacteria and certain viruses don't like to live and can't live in high temperatures. And so this slows down their growth by exposing them to high temperatures. What also the pyrogens are going to do, or fever in general is going to cause, this increase in temperature causes the liver and spleen to sequester or to hold onto iron and zinc. Iron and zinc are highly involved in many different immune processes. So it tells them, hey, save as much iron and zinc as you can so that we can help get rid of this infection. And then also, not only does an increase in temperature or a fever slow down the growth of bacteria and viruses, it can also, as you already know, increases metabolic rate. So it makes cells work faster, including white blood cells or leukocytes. And so then they can fight off these invaders at a faster rate. So a high metabolic rate means faster immune functions.
What would it mean to be sequestered iron?
Huh?
What does it mean to sequester?
Oh, they hold on to it. So they save iron and zinc instead of allowing it to be released in the urine, things like that. They hold on to it. So that white blood cells can use it. Okay, anybody still need these notes up? Okay. So this is just a summary table of all of our second line of defense, all the internal innate defenses, which includes phagocytes, natural killer cells, the inflammatory response, antimicrobial proteins like those interferons and complement proteins, as well as fever. So that was our first and second line of defense. Now we're into the adaptive defenses, which is our third line of defense. And so one thing to know, or a few different terms to know about our adaptive defenses is one, they're specific. So they recognize foreign antigens and respond with matching specific antibodies. So we talked about the specificity of antibodies already with blood typing. So antibodies are specific to a very specific antigen. So all of our B cells, so we have different B cells that produce different antibodies that are specific to different species of bacteria or I had the word a second ago, strains of viruses as well. So they're very, very specific. This is going to be systemic. This is not just a local response. So like with the inflammatory response, that's localized in that area wherever the injury is. If a foreign invader gets past the first line of defense and the second line of defense, now we're in the third line of defense, these get released in circulation all over the body, these B cells and T cells. Okay, so this is going to be systemic and not just in one area. Also, this line of defense has memory. So we have what are called sensitized cells that stay around for months and years at a time so that we can respond more quickly in case we're ever exposed to that particular virus or pathogen again. So that's the whole idea behind vaccines, that you get exposed to the pathogen ahead of time so that whenever you get exposed again in real life, then your body's more prepared to fight that off. And we'll talk about vaccines towards the end of today's lecture. So you'll see here the antibody A that's very specific to this triangular shaped antigen. Here's antibody B that's very specific to this round shaped antigen here. And then antibody C, which is very specific to this irregularly shaped antigen there. Okay, so it's very, very specific antigen to antibody binding. So we divide these adaptive defenses into what's called humoral immunity and cell-mediated immunity. You should already be familiar with the word humoral. We learned about like humoral stimulation of endocrine glands when there's a change in the fluid or in your blood. So a change in calcium concentration or something like that that causes the release of a hormone. So humoral is related to fluid. So humoral immunity is going to be the immunity that we find from fluid in our body, specifically plasma, which is where our antibodies are. So another term for humoral immunity is also antibody-mediated immunity. So you can use either one, but humoral just relates to fluid, specifically plasma, and that's where the antibodies are. So humoral immunity is going to be antibody-mediated immunity, And which of our lymphocytes are responsible for making antibodies? B cells. So humoral immunity is through B cells. Cell-mediated immunity, which means that a cell is attacking whatever the foreign invader is. This is going to be our T cells. And which of our T cells are the ones that fight invaders directly? Cytotoxic T cells. So here's a cytotoxic T cell fighting off a cancerous cell here. Any questions about humoral versus cell-mediated immunity? Okay.
Professor,
Yeah.
the specific type of cell for the B cells, that would be, what would that be?
So we'll talk about this in a second in more detail, but we have plasma B cells. Those are the ones that make the antibodies specifically. And then we have memory B cells that kind of stick around and allow us to have a bigger response if we're ever exposed to the same virus or bacteria or fungi again. Mm-hmm. So you should already know the difference between an antigen and an antibody. Antigen is any recognizable protein. And when I say recognizable, I mean recognizable by the immune system. So any protein that the immune system can recognize. And these can be either self or non-self. Another word for non-self is foreign. So it can be a self antigen or a foreign antigen. But your immune cells should be able to tell the difference between a self cell and a foreign cell and know which ones to attack. And then antibodies are the chemicals produced by our B cells that bind to the antigen so that we could carry out a response. So just like with the blood typing, the antibodies bind to the foreign red blood cells, cause agglutination, clump them together. And when you clump a bunch of red blood cells together, it makes it easy for a phagocyte, like a monocyte or a macrophage, to come and just engulf all those things that don't belong. That's what agglutination is for, and we'll talk about that in more detail as well. So we're going to talk about a specific self-antigen called the major histocompatibility complex. So that's here. So the major histocompatibility complex is a set of proteins on the cell surface that present antigens. It is very, very specific to you, as your DNA codes for a unique protein combination. Only identical twins will have the exact same MHC. So everyone's is going to be different. Okay? And they can actually use your MHC in forensic testing or forensic analysis to link something, some kind of blood that's left at a crime scene or something specific to you. So technically you could set up an identical twin for a crime that you committed, but only an identical twin. Now your family should have very similar MHCs to you. So identical twins will obviously be a really good candidate for a transplant if you need a transplant, because you guys have the same MHC, which is basically a self-identifying protein. So it's what the T cells look for when they're trying to tell something is self or non-self. So in order to prevent rejection of a transplant, it's good to receive it from an identical twin. But it's also good to receive from a close family member as well, because your MHCs will be very similar. Not always exactly the same. Not every family member will be a good match, but a lot of times you can find a good match because the MHCs are so similar. And then T cells have to be able to recognize that self MHC, again, in order to be able to tell a self cell from a non-self cell. That's all part of developing immunocompetence, which is what we talked about last time. And so that's going to be actually their first step in training, is to be able to tell that self MHC and be able to recognize it. So we'll talk about how that works. And yes, you have to spell this one out. You can't abbreviate this one. It's called the major histocompatibility complex. It makes it so that your tissues are compatible with each other. Okay? Your immune cells are compatible with every other cell, and they can tell a self cell from a non-self cell. So major histocompatibility complex. All right, again, we're still on adaptive defenses. We're going to talk about B cells first, and then we'll get into T cells. But as far as B cells and T cells go, this part is review, so you don't need to write anything down. But you should know that our blood cells, including our lymphocytes, are made in the bone marrow during hematopoiesis, or if we're talking about white blood cells specifically, leukopoiesis. But then once they are made, they mature in two different spots. So the B cells will remain in the red bone marrow to mature. That's why we call them B cells for bone. And then the T cells will then travel to the thymus for their maturation. Once they're mature, they've done all their training, and they have developed immunocompetence, then we can put them into circulation, and that's where they would potentially encounter their antigen. Is it a primary or secondary lymphoid organ where lymphocytes encounter their antigen?
Secondary.
Secondary. Okay, primary is where they mature, secondary is where they encounter their antigen. So we put them in circulation. They will encounter their antigen at secondary lymphoid organs, including lymph nodes. Also, we learned about the spleen and the appendix and Peyer's patches and tonsils. But then also, they'll be in circulation as well. So they'll go between the cardiovascular system and the lymphatic system, and cardiovascular system and lymphatic system, and back and forth all the time. When they do encounter their antigen, though, they can make copies of themselves to help fight off whatever that antigen is, whatever the invader is. So how B lymphocytes work is you can see three different B lymphocytes here. Here's one here, one here, one here. But these antigens are only binding to one of these cells. Okay, so you have lots and lots of B cells that you get produced, or that your body produces, and they're all going to be very specific to a very specific shape of antigen. Okay? And so they're not going to get activated until they are exposed to their very particular antigen. Okay? Whether that is Streptococcus A bacterium, or whether that's E. coli, or whether that's the influenza A virus from this year. Okay? So every B cell is going to be very specific to a specific protein or antigen. And then once the antigen binds to that particular B cell, then they can clone, okay, or proliferate. So the antigen binds to the B cell can make a clone, and when they do clone themselves, they're going to clone into plasma cells, which make the antibodies, and then memory cells that kind of just remember that particular antigen and stick around in case you're ever exposed to it again. So you do produce some antibodies the first time you're exposed to something, but then because of these memory cells that we have, if you're exposed to them again, the response is so much bigger. Even more plasma cells, even more antibodies, and even more memory cells. So that if you're exposed to it again a third time, that response is even bigger and bigger and bigger every time you're exposed to it again. Okay. Any questions about that? Okay. So how the antibodies that those plasma cells produce work, it depends on what the antigen is. It depends on what the foreign invader is. So one thing we already learned about was agglutination. So they can bind a bunch of cells together that don't belong there and clump them together. And that clumping enhances or makes phagocytosis easier. Makes it easier for a monocyte or macrophage to come and engulf 10 blood cells at a time than it is trying to catch one at a time. Okay. Antibodies can also cause neutralization. So if there's a particular toxin, let's say botulism, so there's a particular toxin that you're exposed to. The antibody can bind all around that toxin, and now that toxin cannot harm your body. because it's been neutralized or been bound all the way around. Once it gets neutralized like that and antibodies bind all the way around it, then again, that makes it easier for a phagocyte to be able to find it and get rid of it. Just going to go look for the antibodies. The last one, or not last one, the second to last one is precipitation. This is for soluble antigens. And I'll erase this so you can see the word soluble. We'll go back. So precipitation is only for soluble antigens, which soluble antigens are those that are dissolved in the blood. So could be a toxin or a piece of bacterium or something that doesn't belong. But if it's dissolved in the blood, if that foreign antigen, that foreign protein is dissolved in the blood, then it's not easy for an immune cell to pick it out if it's dissolved, right? If you have a cup of Kool-Aid, right, you're not going to be able to pick out the individual red molecules that made that Kool-Aid, right? It's not easy to do. So what antibodies can do is bind to those little soluble molecules, and it precipitates them or makes them come out of solution. So it turns something dissolved into something like an ice cube that's now floating in the blood. And it's easier to identify. So once they precipitate it and it comes out of the blood and it's no longer dissolved, then a phagocyte like a monocyte or macrophage can come in and engulf it. Just makes it easier to see, easier to capture. And then the last thing that antibodies do is they can activate those complement proteins. So we talked about those 20, the group of 20 proteins that can make that membrane attack complex. Antibodies can activate them and tell them, okay, go ahead and make that MAC complex, or go ahead and fight off this particular bacterium or virus, because they do have many different functions and many different ways. We're not going to talk about them all, but antibodies can trigger those complement proteins to start working together to start fighting off whatever the foreign invader is. So there is an acronym to remember all of these functions. So antibodies plan. So they precipitate, they induce cell lysis through the complement proteins. I'll say via complement. They do agglutination. And they do neutralization. So antibodies plan. I didn't say hi. Good morning. Any questions about how antibodies work or any of these processes? Yes, Susie.
Is that the order it has to go?
No, there's no particular order. These are just all their different functions. But it's easy to remember something if you have an acronym for it.
So we say that the complement activation is like giving the green light.
Yeah, yep. All right, T lymphocytes we're going to talk about next. Again, they develop in the bone marrow, but then they mature in the thymus, and then they get released into circulation, and they encounter their antigens at secondary lymphoid organs. So we're going to talk about what happens in the thymus. That's part of their maturation, is that T cells have to undergo training. So they have to be able to develop that immunocompetence where they can tell the difference between a self cell and a non-self cell. So how they do that is through two different tests. The first one is called positive selection. So in positive selection, they have to positively recognize that self-MHC, so that major histocompatibility complex, those set of proteins that are on the surface of every one of your cells, the T cells have to be able to positively recognize it. They have to be able to bind to it in order to tell, okay, I recognize this thing. This is a self cell. I can ignore this cell. It's not harmful. I don't need to attack it. So you can see here, this T cell was able to, so this is a developing T cell over here. This is a thymic cell over here with an MHC. So this is the MHC here, and this is the T cell receptor. And you can see that there, T cell receptor on the blue side, self MHC on the other side. Right here at the bottom, you can see that they're properly bound together. Everything matches up. Okay? And so if that happens, recognizing the self MHC results in survival, and then the survivors can proceed to the next test. So once they pass this test, they move on to the next test. If they cannot recognize it, you can see they do not match up perfectly here. They have not positively recognized this particular self MHC. And so what ends up happening is that cell will then undergo apoptosis. So regulated cell death. That T cell is non-functional. It cannot tell a self cell. It does not bind properly, so it must die. Because you don't want to have an autoimmune disorder where a T cell doesn't know what to attack and what not to attack and starts attacking your own tissues. So any T cell that's not properly trained and cannot recognize this self protein will then undergo apoptosis and die. Pretty cutthroat.
Yeah, I was just thinking, open your eye and see.
Right? There's no like, I half recognize it. Nope, you can't recognize it, you're gone. All right, so that's positive selection. They have to be able to positively recognize that particular self MHC for that person that they're living within. So the next test, so if they do recognize that self MHC and they're able to move on to the next test, that next test is now called negative selection. Okay, and so the purpose of this test is this is the self MHC. This is a different self antigen. So when a cell or a T cell mounts a response, it fully binds to whatever the MHC is and that self antigen. It fully binds, okay, when it's going to mount an attack. So we do not want a T cell to be able to fully bind to our cells because then they'll want to attack it. So this negative selection is, yes, they can recognize the self MHC and they can bind to that part, but we do not want them to fully bind to this other self antigen. So you can see on this one, a little room is left, so it's not fully bound. So that means that T cell is not going to attack that particular cell. Okay. You see over here on the top, it is fully bound. It recognizes the self MHC and it binds to the self antigen. And that means that T cell is not going to attack that cell. So if it cannot negatively select or not fully recognize that particular self antigen, then again, they're going to undergo apoptosis. Okay? We do not want them to mount a response against our own cells. So we do not want them to fully bind. Okay? So for positive selection, yes, recognize the self MHC. For negative selection, no, do not recognize the self antigen. Okay? So if they fully bind, they undergo apoptosis, that T cell's dead. It did not complete the training. It's not going to be useful for our immune system, so we get rid of it. If it's able to leave a little bit of space, okay, just like at a junior high dance where they tell you leave a little bit of space. Okay, if they can leave that little bit of space, that means that they are going to survive and continue on with their maturation. Okay. I'll give you a little clue about how an exam question on this might go. Okay. So It's like a cascade of events. So first we have positive selection, where we want recognition of the self MHC. Okay. So if the T cell can positively recognize that self-MHC, they are going to survive and proceed to negative selection. If it can't recognize that self-MHC, then apoptosis. From there, negative selection is all about recognition of the self-antigen. But this is negative selection, so we don't want it to recognize that self-antigen. So again, negative selection has to do with the recognition of the self-antigen, but it is negative selection, so we do not want them to recognize it. So if they do recognize it and fully bind to it, apoptosis. But if they're able to leave a little space and do not fully recognize it, do not fully bind to it, then they can survive and complete their maturation. So you should be able to tell this story on your next exam. Does that process make sense to you all? Any questions about how that works? Okay. Anybody still need this up? Because I'm going to make it go away. Okay. What happened? Did it pop?
No.
Oh. Oh. These chairs are not comfortable.
It's my dog's fault, actually.
Really?
Yeah, she likes to sleep like right in the nook of my back and has no.
Oh.
She has no personal space.
Yeah. Okay, anybody still need this up? Okay, it's going away in 3, 2, 1. Okay.
She's actually a German shepherd.
Oh gosh.
Like right in my back all night.
Okay, a little bit more about T cells. You do not have to know this huge diagram. Just take away the bullet points. So helper T cells, they do form memory helper T cells. Okay, so these are ones that stick around for years to make sure that we're ready when a foreign invader comes to us again. Why they're called helper T cells is that they help with the activation of B cells and they help with the activation of cytotoxic T cells as well. Okay, they help activate. That's their job. Okay. They're more of an immune messenger than an actual like fighter in the immune system. And so you don't have to know these processes either, so don't freak out about all the different things that are involved. All this is here to show you is that this helper T cell helps activate both the B cell and the cytotoxic T cell. Okay? Again, you don't need to know the steps. If you want to learn about it, go ahead and read in your textbook. I think I go into it a little bit more detail in the online lecture that's posted, but you're not going to get tested on the details. So just know helper T cells are going to activate both our B cells and our cytotoxic T cells. So cytotoxic T cells, they also form memory cells. So a big thing that's this adaptive immune system is that it has memory. It can remember past infections. So they form memory T cells. They attach and lyse things with a foreign protein on it. So this includes tumors, which is a good thing. We want our cytotoxic T cells to be able to fight off cancerous cells and tumors. Okay, but then also it's cytotoxic T cells that will fight off a transplanted organ, which is not a good thing, which is why when people do get transplants, they get put on immunosuppressive therapies so their immune system and their immunity is lowered so that they have a higher chance of accepting that transplanted organ. Yeah.
Are you on immune suppressants for like the rest of your life?
That I don't know. I know for years after you get that transplant, because you just never know at what point the body is going to reject it. That girl from Gossip Girl that just died, she had just recently had a liver transplant, I think over a year ago. They think that it was complications from her rejecting it, which is very sad. So I imagine for years ahead, but I'd have to look it up to see. I'm sure it's different for different cases. So how these cytotoxic T cells work. And let me see if these steps are in your notes. Yeah, okay. So here's a cytotoxic T cell. Here's the target cell. Could be a transplant cell or a cancerous cell. Or it could be a virus. But it binds, the T cell, cytotoxic T cell, binds tightly to foreign cell. And it releases two different chemicals. It releases perforin, and these are proteins that create pores that are similar to the membrane attack complex that we talked about earlier. They're just a different set of proteins, but they're still making a pore. And they also release a chemical or an enzyme called granzymes. And these are going to trigger apoptosis of the target cell. So basically what happens is that the cytotoxic T cell creates these pores in the target cell, releases the enzymes so that the enzymes can go through the pore and into the target cell, and then it causes the target cell to then undergo apoptosis and die from the inside. And it does it by activating certain enzymes on the inside of the target cell. So questions about how cytotoxic T cells work. Okay. You don't have to know things about like MHC1, MHC2, so you can just go with the summary that I wrote up instead of the exact steps that are listed in the textbook. Because we only have one lecture on this chapter, so we can't really get into a whole lot of details. So I try to give you as much as I can with staying on the surface level. So here's cytotoxic T cell. This is a micrograph, so an actual microscope image of a cytotoxic T cell attached to a cancer cell. And again, releases the proteins to create pores, releases the enzymes into the target cell or this cancer cell here and causes apoptosis of the cancer cell. We already talked about the HIV virus. or HIV last time. It replicates inside of helper T cells, destroys those helper T cells, and helper T cells are needed to activate B cells and T cells. So if we don't have those helper T cells, we don't get activation of our actual cells that do the work, and so the immunity gets lowered. And so what does HIV develop into? AIDS, okay, or acquired immunodeficiency syndrome. And so what ends up happening with AIDS patient is that they die from infections that are not usually lethal. So they'll die from bronchitis or pneumonia or something like that because their immune system function is so lowered that their body can't fight off these simple infections that any normal person would be able to survive easily. Or they die from cancer also much more quickly. So next is active versus passive immunity. So we're talking about like vaccines and such here. But this is all done through humoral immunities. This is all antibody related. So active versus passive. So active is when you make antibodies yourself. This is active immunity. Passive immunity is when the antibodies are passed on to you. Okay, so active immunity, you make those antibodies. Passive immunity, you acquire those antibodies. And so we'll go into active immunity first. Oh, you want to write that down really quick? Everybody have those two little definitions right now? Okay. So active immunity, when you make the antibodies yourself, that active immunity can either be naturally acquired. That means that you actually were exposed to the pathogen or the antigen in real life that caused your B cells to make the antibodies. That is naturally acquired active immunity. Artificially acquired is when you're exposed to that antigen through a vaccine. You weren't actually exposed to it in nature. You were given a vaccine that caused your B cells to have that same reaction, but you're still making the antibodies yourself. Whether you were naturally exposed or exposed via vaccine, you are still making those antibodies. Okay, but we just divide them into naturally acquired versus artificially acquired. And with the vaccine, you're usually either given a dead or weakened version of that pathogen, so you don't really get infected. You might have some symptoms of being sick, but you're not getting a full infection because that particular virus isn't really harmful. It just causes your body to have a reaction. Or with like the COVID vaccine, it's an RNA virus. So it actually causes your cells to make the protein that looks like COVID, so you still have that reaction. And your B cells still make the antibodies against it. Passive immunity is when you acquire the antibodies, you don't make them yourself. Naturally acquired is when the antibodies are passed from mother to fetus, either through the placenta or through breast milk. So you don't make them yourself as an infant, you're just passed on to them, or your mother passes them on to you, either through breast milk or the placenta. And then artificially acquired. This is when you have an injection of antibodies. So some people are given antibody cocktails for certain infections and illnesses, and that would be artificially acquired. So passive immunity is when you don't make the antibodies yourself. They are naturally acquired if they were passed on from mother to fetus or mother to infant. Artificially acquired if you're given an injection of antibodies. Everyone understand these categories? Okay. Why vaccines work? Again, it's because your secondary response is always going to be bigger than your initial response. So if you are given a vaccine, your body already knows what to look for. It already knows what that particular pathogen is. So if you're ever exposed to it in nature and in real life, then you can have a much larger response. You can see this diagram here. This is antibody titers in different concentrations here. Here's the first exposure. And then look how much bigger the second exposure is. You have a much bigger response, much more likely to fight off the infection quickly so that you don't get such severe symptoms. Okay? But for every new antigen, you start all over. Okay? Because your B cells are specific to very specific antigens. So this process is going to be different for every different specific antigen. Questions on that? Okay. This is just a summary of all those different parts of the immune system that we talked about, and this just shows you how complex our immune system is. There's many different facets to it, lots of different protections that we have to help us fight off any foreign invaders. So homeostatic imbalances, really quickly, immunodeficiencies, it can either be acquired, like with HIV, that turns into AIDS, or it could be congenital, where you have white blood cells or antibodies that are not working properly, like when we talked about like leukemia, or we'll talk about type 1 diabetes, also an autoimmune disorder. But these are, again, immunodeficiencies, and they can either be acquired or you can be born with them. Immunosuppression is when you have a decreased immune response. This can either be drug-induced, like with chemotherapy. Chemotherapy is toxic to cells, and not just the cancerous cells, but all your cells, including your immune cells too. So during chemotherapy, you will have a drop in immune function as well, as your white blood cells are being attacked. Disease can cause a decreased immune response as well. But then it also can be intentional. So we talked about immunosuppressive therapy, where you can take corticosteroids to suppress your immune system, to lower your chance of rejecting a transplanted organ, or to decrease inflammation. Maybe you have rheumatoid arthritis, so you have lots of inflammation going on. You can take certain anti-inflammatories to help with those symptoms. Yes, it will lower your immune system, but your immune system is what's causing the rheumatoid arthritis anyway. Anti-proliferative drugs, which stop white blood cells from being produced, like with the case of like leukemia. These are all going to be intentional though. Autoimmune disorders, this is when your body fails to recognize self proteins versus non-self proteins. And so then it unleashes the immune system on your own cells. You're attacking your own cells. So this includes multiple sclerosis, which you learned about in Bio 32, where the myelin sheath is being attacked, and so signals are not being sent the way that they should. With multiple sclerosis, you go through times of relapse and remission, so it gets better and then gets worse and goes back and forth. We talked about Graves' disease, where you get exposed to a certain environmental trigger that causes you to make these abnormal antibodies, which is causing you to produce more thyroid hormone than usual, increasing your metabolism and causing lots of different effects, including the bulging eyes or exophthalmos. Rheumatoid arthritis is when your immune system attacks your joints specifically. Lupus is system-wide, so lupus is very unfortunate autoimmune disorder that can attack many different organs of the body. Type 1 diabetes is when your pancreatic cells are not working properly, and so they are not producing insulin the way that they should. So this is a hyposecretion of insulin. So if you're not making enough insulin, more glucose stays in the blood, causing lots of different symptoms, which we'll get into with the digestive system. This is type 1 is autoimmune, type 2 is not. Type 2 is due to lifestyle and insulin resistance, so we'll talk about that a little bit later. Type 1, though, is autoimmune. And then glomerular nephritis is when your immune system attacks your kidneys specifically. If the damage is already done, then there's no treatment to prevent it. Like with type 1 diabetes, you just might need to be put on insulin to help with the symptoms. But if the symptoms are ongoing, then you can be put on immunosuppressive therapies to help decrease your symptoms. Last thing really quickly before we get out of here, hypersensitivities. This is when your body harms itself because it thinks something is harmful when it's not. So it causes an immune response to things that are not harmful, but it thinks it is. Like cat dander. I'm allergic to cats, and it's not dangerous, but my body thinks it is. Or grass, or pollen, or whatever it is. Allergies, though, are a mild form of this, where you get stuffy nose and maybe scratchy throat, things like that. You can take antihistamines or corticoid creams to stop with allergies. But anaphylaxis is a much more severe response in which you can have swelling of the tongue, and it might be hard to swallow, maybe hard to breathe, because you can get swelling of your throat tissues and you can't get air ventilation to occur. It can cause hives, can cause loss of consciousness due to the lack of airflow. And so anaphylaxis is a much more severe response than just allergies. And that's when you might need like an EpiPen or something like that to help with those symptoms in an emergency situation. Don't forget about your PLA on the respiratory system, respiratory anatomy, that is due on Wednesday. It needs to be either flashcards or a Quizlet or notes on those things, plus the diagrams labeled. If you need another copy of it, I can have some in the lab for you. No lab today unless you want to come review respiratory ahead of time.